everyone!
Today's topic is about tissue microscopic observation in histological techniques. It is about observing the stained tissue specimen under a bright field light microscope. What is a bright field light microscope?
Bright field microscope, also known as a compound light microscope, is an optical microscope that uses light rays to produce a dark image against a bright background. It is the standard microscope used in many labs. Specimens are first prepared through staining, which adds color to facilitate clearer contrast and characterization. Once stained, the specimens acquire a refractive index distinct from their surroundings, producing both absorption and refractive contrast. This is an example of a tissue slide after the staining process. The dye used during this process are hematocillin and eosin. Right after staining, the tissue will be mounted with a cover slip using DPX. Now that the staining and mounting part is done, let's move on to the fun part.
First, turn on the microscope. Then, place your glass slide on the stage and secure it using the metal clips. Adjust accordingly using the coarse and fine adjustment knobs.
If you can't see anything, try checking your light source or the diaphragm and make sure it is not fully closed.
Under 4 times magnification, the tissue are stained in reddish and pink color due to the eosin dye. The nucleus are not visible yet. Under 10 times magnification, the image is now clearer, and the tiny purple dots, which is the nucleus stained by the hematocillin, is now visible.
The white spaces you see were results of mishandling during tissue crossing or processing.
Under 40 times magnification, the hepatocytes and the nucleus can now be seen individually. This is a clearer, nicer image of hepatocytes under the microscope, which were taken from another source. We can see the sinusoids, which is a small irregularly shaped blood vessels often found in the liver. We can also see the central vein, which is lined by a single layer of endothelial cells.
But wait, have you ever think about how the dyes know what to stain and what not to stain? How does it work?
Hematocillin is a positively charged basic dye with a purplish-blue color. It stains acidic or basophilic structures such as the nucleus, which contains DNA, and organelles that contains RNA, such as ribosomes and rough endoplasmic reticulum. Eosin is an acidic dye that is typically reddish or pink, and it stains basic or acidophilic structures, including the cytoplasm, cell walls, and extracellular fibers. It is used to distinguish the cytoplasm of different types of cells and different types of connective tissue fibers. It is also the most suitable dye to combine with an alum hematocillin. For routine histological work, 2 dyes are used for a primary stain and a counter stain. In this case, the primary stain are hematocillin that stains the nucleus in bluish color, and eosin being the counter stain as it stains the cytoplasm and other organelles in reddish pink.